So this is video 3 of proteins. So some of the things that I want to cover are coenzymes, cofactors, um, things like the heme inside of hemoglobin. I want to talk about enzymatic pathway regulation, so allosteric changes or conformational changes. How do we prevent toxic buildup by regulating the beginning of the pathway? We've already talked about that a little bit. And talk a little bit about post-translational modifications like how do we turn proteins on and off, like adding a phosphate group via a kinase or GTP binding proteins that will then hydrolyze the GTP to GDP and that way turn proteins on and off. Some stuff about protein complexes and proteolysis. And then maybe talk a little bit about some of the techniques that we use to study proteins. So, so like I said before, we really don't like for protein to have any hydrophobic amino acids exposed to the water aqueous environment of the cytosol. So as the protein is being made, um, hydrophobic amino acids are going to be in the primary structure. And so how do we deal with that is that chaperone proteins or heat shock proteins are going to bind to those hydrophobic amino acids. In this case, we can see that as the protein is coming out of the large ribosomal subunit, uh, HSP70 or heat shock protein 70 kilodaltons is going to bind to those hydrophobic amino acids and make the primary structure soluble. So now that it's in the cytosol and it's soluble, it can do one of two things. One, it can fold up correctly, maybe creating some alpha helices, beta sheets, and then the overall globular tertiary structure of the protein. Or B, that doesn't happen and it has to go into a chaperone pathway. So an unfolded protein creates something called the unfolded protein response, which is an upregulation of chaperone proteins, heat shock proteins. They were first named heat shock proteins because if you heated up a cell, then proteins would unfold, and so then these proteins would be part of the unfolded protein response. They would be upregulated in response to this heat shock. And so that's how they were originally named, and that's why many of them are called HSP. So the HSP60 is kind of like a little can that has inside of it a microchemical environment that is more conducive to the protein folding up. Less water and uh, um, a reducing environment or an oxidizing environment instead of a reducing environment. And and so we keep doing this chaperone protein, try to refold it, chaperone protein, try to refold it until it gets refolded. If the protein never gets refolded, then the cell might have some really consequential problems. I mentioned in the last video the single base pair change in the hemoglobin protein that causes the substitution of a glutamic acid, which is polar, for a valine, which is nonpolar. And that valine being exposed to the aqueous environment is not happy at all. And so it binds to another valine on a different hemoglobin. And so then you get these large aggregates because, of course, hemoglobin is in a quaternary structure normally, and so you would have a hemoglobin on either side of the quaternary structure that it has an exposed valine. And so then those those hemoglobin complexes will bind to each other on either side, and there will always be another valine on the other side. And so you get these large aggregates of protein that cause a change in shape of the cell. And then that change in shape doesn't allow these red blood cells to go through the capillaries, and so you get clogging up of capillary beds, and that's going to cause destruction of organs and ultimately organ failure due to hypoxia. But in the meantime, it causes a lot of pain for the people who have sickle cell anemia because their muscles are always starved for oxygen because the clogging up of the capillary bed. So um, uh, another example of these insoluble aggregates is in the case of spongiform encephalopathy. So spongiform encephalopathy is also widely known as mad cow disease, but mad cow disease is only one form. It's the scrapie form in cows, bovine encephalopathy. Um, so bovine spongiform encephalopathy is just one form. There's also scrapie in sheep. There's kuru among some cannibalistic tribes in Papua New Guinea who eat their tissues of their ancestors, and then there's also a genetic form called Creutzfeldt-Jakob's disease. And so all of these spongiform encephalopathies have one thing in common, and that's the uh, spongiform form of the protein gets misfolded, and when it gets misfolded, it exposes a whole bunch of insoluble amino acids to the surface, and so then you get these large insoluble protein aggregates inside of neurons, and then the neuron dies, and that's how you get the the encephalopathy in the case of all of these scrapie diseases. This was originally discovered by Stanley Prusiner, and he called it prion disease because it was a protein that was infectious. If he gave this protein from the scrapie sheep to another sheep, then that sheep would get the spongiform encephalopathy just like the original sheep. And so this scrapie form of the protein was actually what was causing it. It was the normal protein, which is it's a copper binding protein. The normal form of the protein would get transformed by the spongiform form of the protein. And so then once it transformed the first protein, then that one would also be able to go on to transform other proteins. And and that's how the disease is propagated. Stanley Prusiner eventually won the Nobel Prize in 1997, but not without some controversy before he won it, because of course this flew in the face of the DNA to RNA to protein central dogma. And so, if you're going to overturn something called the central dogma, you're going to need a lot of evidence. And once he had finally amassed all of this evidence, they were laughing at him until he won the Nobel Prize. So, this is just a picture of uh, the holes that are generated due to the dying of neurons, and you can see plaques depositing from these prion proteins, in insoluble prion proteins, creating these plaques that cause the neurons to die. So I also wanted to talk a little bit about regulation of a pathway, and so I'm going to stop this video and allow you to go ahead and try to find a um, try to find a good regulation uh, of this biosynthetic pathway. So A goes to either F or H. And so I want plenty of F, plenty of H, and no toxic buildup of E, G, D, B, or C. So stop the video and try to do this. Okay, so I guess you're back now, and so the answer is if F inhibits enzyme 4, H inhibits enzyme 6, and D inhibits enzyme 1, then you won't have a toxic buildup of E, G, D, C, or B, and you'll have plenty of F and plenty of H. If there's too much H, it inhibits enzyme 6, and then D will build up and just go down the pathway of EF. If there's too much F, then it will inhibit enzyme 4, and then D will go to GH. If there's too much F and H at the same time, then both 4 and 6 are going to be inhibited. So then there will be a buildup of D. Well, we didn't want a buildup of D, so if we then have D inhibit enzyme 1, then there won't be any buildup of B, C, or D until there's not any more D, um, and that would relieve the inhibition and start the pathway again. So this was just a review on that uh, uh, control of enzymes. So this concludes video 3. So thanks for watching.